Okay. Hello everyone. First, thanks to the organizer for giving us this opportunity to share some of our work. At Ashoka, as a research group, we are currently trying to understand the impact of diet on phenotypic plasticity and evolutionary adaptations of a number of traits which are at the level of organism as well as at the suborganismal level. This particular study is a single generational study where we are trying to understand the impact of dietary macronutrient composition on dietary macronutrient composition at the level of during their developmental time, developmental phase, as well as during the adult stage on reproductive output. This is important simply because reproduction or reproductive output is a key component of evolutionary success of any organism. However, no organism starts reproducing at birth, right. So, before reproduction they develop and grow and then they become reproductively active. During this pre-reproductive and developmental phase also they need food, right. So, during both the stages they need food, but the dietary requirement of these two kinds of food might be different. So, therefore, this led us to ask this question that how does this stage specific diet composition can affect dietary output in organisms. However, we also know, and also it has been shown experimentally, that reproductive output of an organism does not stay constant throughout its life, it changes. It also undergoes age dependent age related declines, right. So, we refined our questions to this objective, the first one, that is what is the effect of or what is the effect of developmental and adult diet and their interaction on reproductive output across age, and also you would like to see what is the mechanistic basis or physiological basis of those effect what we will be seeing as a result of the objective 1. For our study we have used one of the most popular model organism, which is Drosophila. Relevant to this particular study, the Drosophila undergoes this 4-stage life cycle where we have this larval stage where they are heavily dependent on food. However, that food is primarily used for growth and development. Around if we consider the date of egg collection being the 0th day, around 9.5 to 10th day we get most of the flies closing out of pupal stage, when again they need food, right, and they also reproduce at that stage. So, we will be dividing this life cycle into developmental phase and adult stage, and food available for those two will be referred as developmental diet and adult diet. The experimental population is started with a large outbred population which we call baseline population, which are maintained on baseline diet, which has a protein is to carbohydrate ratio which I mentioned here as P is to C to be 0.4. We collected eggs from those populations, baseline populations, and reared them under two contrasting diet conditions with high protein diet, high protein diet which has a protein is to carbohydrate ratio of 0.7 and low protein diet which has a protein is to carbohydrate ratio of 0.25. Remember that all the diets including the baseline diet and this high protein diet, low protein diet, they are all of isocaloric nature. Whatever flies, whatever adult flies we got from each of these two treatments, we further splitted them into two parts. We gave high protein diet as adult for the adult diet and low protein diet as adult diet. So, we have in total 2 by 2 4 combinations with high protein and low protein as developmental diet and adult diets. And this is a very nice framework because we could easily dissect out using generalized linear model framework the main effect of developmental diet, adult diet, as well as dissect out their interaction if there is any. So, what did we get? Okay, before that we also have to remember that we wanted to also see how diet affects reproductive output across different age points. So, we have these 4 different diet regimes, and for each diet treatments we measured the reproductive output as viable progeny count on 4 different days, day 10, 12, 22 and 32 from the day of egg collection. In each day, we hosted 2 male-female pairs per vials and such 30 pairs, 30 vials per treatment, and we allowed them to lay eggs for 24 hours. We discarded the adults after 24 hours and reared the eggs laid under standard laboratory conditions and counted the number of viable progeny emerging out of each fly, and then we scaled by each female to calculate viable progeny count per female. So, here we will be referring to this day 10 as very early life, day 12 as early life, day 22 as mid life, and day 32 as late life. So, what did we find? So, here this is the result. The way I am showing it is that we are showing on the y axis viable progeny per female. In the x axis is the age, day from the egg collection, the 4 different age points, and there are in the left hand side there are this is for the red ones correspond to the flies which were reared on high protein developmental diet irrespective of their adult diet, and the blue ones were reared on low protein developmental diet irrespective of their adult diet. And similarly on the right hand side, it's essentially the blue one, the red one is essentially got high protein adult diet, and the blue one got